In this video, we're going to look at how we use some biotechnology so that we can have bacteria clone a gene. So before we start, I want to just look at a few different definitions of some terminology. So genetic engineering is when we actually take genes from one organism and put them into another organism. So in our gene cloning example, we're going to use a human insulin gene and we're going to put it into bacteria. And we're going to make bacteria produce insulin that we can actually use to treat human diabetes. When we look at some other definitions though, so biotechnology itself doesn't necessarily mean that it has to be genetic engineering. So biotechnology could be something like using yeast to make alcohol or bread. When we say genetic modification, we can mutate the genes within a cell. So let's suppose in a lab you have a cell line, a cell culture, and you want to specifically mutate one of those genes. Say you want to turn off a gene or change a nucleotide sequence so the protein is dysfunctional. Okay, you're not inserting any new DNA, you're just changing the DNA. So that's genetic modification. A GMO is a genetically modified organism. So this is when you can alter the DNA or put new DNA into an organism like a plant or an animal. And then lastly, we have selective breeding. And this is not genetic engineering. Selective breeding is artificial selection. So this is when humans decide which organisms are going to breed to produce the next generation. And we do this with plant foods all the time. And same with chickens. You know, the chickens that lay the most eggs, the cows that make the most milk. Okay, then the farmer chooses which organisms. So that's selective breeding. So this video, we're going to focus on genetically engineering bacteria so that they can produce a human protein. Genetic engineering is actually a little bit like cooking. So you have to have a recipe and you have to have ingredients, and then you have to follow the steps in the recipe, and then you get a product in the end. So this is kind of the same thing. So when we're going to clone a gene, we need certain ingredients. So the ingredients that we need is, first of all, the gene that we want to clone, our gene of interest. So we need our gene. Then we need to have enzymes, special enzymes called restriction enzymes that will cut DNA. And I'll talk about those a little bit more in a second. Then we need another enzyme called ligase that glues DNA. And then we need a vector, something that's going to carry that human insulin gene into the bacterium. And so we use vectors. There are a few different kinds of vectors. The main vector that I'm going to focus on in this video is a plasmid. So then plasmids can go into the host cell, which is our last ingredient, where the host cell, which is our E. coli bacterium, can now replicate the gene of interest every time it replicates itself, and then it can transcribe and translate that gene and produce a protein. So first I want to explain a little bit more about how restriction enzymes work. Restriction enzymes are protein molecules that cut DNA sequences. Now they cut very specific DNA sequences that are called palindromes. So a palindrome is something where you can read it in one direction and it's the same thing if you read it in the other direction, like race car or noon, or was it a car or a cat I saw? So when you can read it in the same, it says the same thing in both directions, that is a palindrome. So here we have an example of a restriction enzyme that we're going to call EcoR1. And this is the sequence that EcoR1 will recognize. But now remember that DNA is double-stranded. So the opposite strand is going to be C, T, T, A, G. So now, how is this a palindrome? When we read the top strand in this direction, we have GAA, TTC. When we read the bottom strand in the opposite direction, we have GAA, TTC. So this is a palindrome. Same with our second example here. If we read this strand of DNA in this direction, we have GGA, TCC, And if we read it in this direction on the bottom strand, we have GGA, TCC. So they are palindromes because they read in the same way in both directions. So an interesting thing about how restriction enzymes work. So now there's going to be a protein, and when it cuts the DNA, it cuts it unevenly. It's going to cut it here and here. So now what that does is it causes sticky ends to be formed. So if we look more closely at what the EcoR1 restriction enzyme cut site looks like, the enzyme is going to come and cut the DNA in this jagged, uneven way. So now when those pieces of DNA come apart, we have sticky ends. This means that now we can insert another piece of DNA. Let's suppose this is the human insulin gene. And if we cut the DNA surrounding the insulin gene with the exact same restriction enzyme, then we will have the exact same sticky ends. So then when you mix those together, they will automatically match up because A's and T's always combine and C's and G's always combine. So now when these molecules come together, we can seal these little sugar phosphate bonds, the phosphodiester bonds, using the molecule ligase. So we can combine these pieces of DNA from two different species, a human sequence and a bacteria sequence, and we form a recombinant DNA molecule. So restriction enzymes are extremely useful because we can cut and paste DNA, and because every living organism, from bacteria, plants, fungus, fish in the ocean to humans, all living things have DNA made of A's and T's and C's and G's. And so if you can cut gene sequences from one organism, you can cut them from another organism, and then you can mix them. So we need restriction enzymes to cut it, and then we need ligase to seal the gaps and form phosphodiester bonds. Now that we have a piece of DNA from our human sequence, our insulin gene, and we put it into our bacterial plasmid, I want to talk just a little bit more about the plasmid. So the plasmid is a vector, and because bacteria normally have plasmid molecules, it's fairly easy to remove the plasmid without hurting the bacteria. We can alter it, put our human gene sequence in there, and then we can put that plasmid back into the bacterium. And then the bacterium will accept it because it's a normal molecule for bacteria, except now it has this extra gene. And then when the bacterial cell replicates, it will copy the plasmid, just like normal, and then we can make millions of bacteria that have our human insulin gene. So a little bit more detail about vectors. There are a few different kinds of vectors, so I just want to take a little look at those before we move on. For a vector to be useful to a genetic engineer, it has to have a few characteristics. Okay, vectors need to be able to self-replicate within the organism, within the host cell organism. It needs to have an origin of replication so that it can replicate. If the plasmid didn't copy itself every time bacteria divided, then you would just end up with a population of bacteria and only one of them would have your gene. Okay, so it has to replicate. It has to have restriction enzyme cut sites so that we can cut the DNA, the plasmid DNA, with the same restriction enzyme as our human gene, and then we can match them up and insert them. So vectors, like the plasmid we're going to look at, has a region called a multiple cloning site that is full of all kinds of restriction enzyme cut sites. So we can choose any enzyme that we want that's going to work with our gene to cut the plasmid. It needs to have a promoter region so that that gene can be transcribed and then translated into a protein. Okay, if we can't make that insulin gene be transcribed and translated into an insulin protein, then it's not going to be a useful medication. So it needs a promoter. And then it needs to have selectable markers. And I'm going to talk more about this when we go through the stages of actually cloning a gene. But let's have a look at what a typical plasmid might look like. Bacteria, if you recall, they have a single circular chromosome. This is their main DNA chromosome. And then plasmids are these little extra pieces of DNA that can be found inside some bacterial organisms. If we remove this plasmid, and now keep in mind that plasmids have been genetically engineered to contain all of the features that make it very useful. There's all kinds of different plasmids that can be used. If we look at a typical plasmid that is useful for genetic engineering, it has an origin of replication, so the bacterial cell can copy and replicate this whole entire plasmid. It has a promoter region, so that it can transcribe our gene that we insert. It has a multiple cloning site. MCS is a multiple cloning site. This is where the restriction enzyme cut sites are located. So we'll have a whole bunch of different restriction enzyme cut site sequences, palindromes, within this multiple cloning region, so that we can always cut the plasmid in this region. We don't want to have a plasmid with a restriction enzyme site over here, and then we end up inserting the gene too far away from the promoter. Okay, so we need to have the promoter upstream of the gene so that it can be transcribed. Now, two other features. We need to have selectable markers. The first kind of selectable marker is antibiotic resistance. So this has AMP, so that's short for ampicillin, which is a type of antibiotic, and resistance. So it can be tetracycline resistance or amoxicillin or any kind of antibiotic resistance gene. So this means that when bacteria contain this plasmid that has this resistance gene, that means they can grow on a plate containing this antibiotic and they won't die. So this is an important selectable marker because only bacteria that contain the plasmid will survive on the plate. Now, our second selectable marker is called LAC-Z. So we've talked about the lac operon in a previous video. The link is below if you want to refresh your memory. And LAC-Z is one of the genes in the lac operon. And we can use this gene as a tricky little way of selecting for bacteria that contain our human insulin gene in the multiple cloning site. So LAC-Z is a normal gene sequence that when you feed the bacteria a substance called X-gal, which is kind of like a fake lactose. So the bacterium thinks it's lactose, so it's going to want to transcribe one of its lactose genes. But if we feed it X-gal, this is actually an enzyme that will make the bacterial colony turn blue. So when this gene is transcribed, when there is no human insulin gene inserted into the multiple cloning site region, so this whole area is intact, then the bacterial colonies are going to be blue. So think about what might happen to that LAC-Z gene if we insert a human insulin gene into that multiple cloning site. Okay, we're going to come back to that. So we'll get to our stages of cloning a gene. Now, just a few other vectors that can be used in genetic engineering for cloning a gene. So plasmids are probably the easiest and simplest to use, but they can only hold a certain amount of DNA. There's other vectors that we can use for other purposes. Another example of a vector that can be used is a bacteriophage. Remember that bacteriophages are virus particles that can infect bacteria. So their DNA, the genetic material in that bacteriophage capsule, can be injected into bacterial cells. So that can be a mode of transmission of getting foreign DNA into that bacterial cell. The process of moving a plasmid into a bacterial cell is called transformation, okay, because bacterial cells can take up DNA from its environment. And the process of moving DNA into a bacterial cell through a phage, a bacteriophage, is called transduction. We can also use what's called a plasmid. So some phages will have what's called a COS site, and this part of the DNA helps to package virus particles. And then you can combine that virus feature with a plasmid, and then you have a plasmid. So then this means that you can actually put more DNA into that vector, and then it will package into virus particles that can be used to infect other bacteria with that DNA. And then the last type I want to mention is a YAC, or a yeast artificial chromosome. So yeast are fungal organisms. They're single-celled fungi, and they are eukaryotic. So remember, when we look at bacteria, their normal DNA doesn't have introns. They don't have RNA processing. They don't have mitochondria. They don't have membrane-bound organelles. So they can't process RNA, and they can't modify proteins after the protein has been made. So sometimes yeast cells are useful in using a YAC because then it's a eukaryotic organism. So eukaryotic organisms can process RNA, and they can modify proteins after translation. Okay, let's go back to our cloning a gene ingredients. We need a gene of interest. We're going to use insulin, human insulin. We need restriction enzymes. We need ligase. We need to have a vector. We're going to use a plasmid. And then we need to have a host cell. We're going to use bacteria. We're going to use E. coli bacteria because they will take up the plasmid. Now, how do you know which restriction enzyme to use? Okay, we can look at a restriction map. Because of the Human Genome Project, we know the sequences of the whole entire genome. So we know where the insulin gene is, and we know all the restriction enzyme cut sites that are within or surrounding the insulin gene in humans. So for example, let's suppose this region of the DNA is our human insulin gene. We can see using a restriction map that these restriction enzyme cut sites exist within and around our human insulin gene. So which restriction enzymes would we want to use to cut out our insulin gene? If we used SMAW1 and BAMH1, what would happen to our insulin gene? We would chop it up, right? We want to use restriction enzymes that are outside of our insulin gene. So we would use HIND2 and BAMH1 in this example because these restriction enzymes are not going to cut up our gene. So once we've chosen our restriction enzymes that we're going to use to cut out our human insulin gene, we have to cut the plasmid with the same restriction enzymes, okay? So that the sticky ends will match, and then the DNA will insert into the plasmid. So any gene is going to have a different restriction map. So you will need to look at a restriction map to figure out which restriction enzymes are going to work for your specific gene so that your gene is intact. Now let's suppose we're going to cut our plasmid with EcoR1. Then we cut our gene out with EcoR1. We can use two enzymes. This could be HIND3 and BAMH1. And then we would just cut with both of those enzymes in our plasmid. And then we simply mix the cut gene of interest and the cut plasmid. We mix them together. You incubate them, and they will automatically find their sticky ends that match. And then we add our ligase to seal the gaps. Now we have our recombinant plasmid. The other thing that I want to mention is that sometimes the plasmid will just fold back on itself. It doesn't always 100% of the time accept the DNA fragment. So this is also why we need to have selectable markers. We are going to use the human insulin gene. Now, we don't have to isolate our gene from a cell anymore. We can actually just order our insulin gene from a biotech company. So you don't have to actually isolate genes from cells anymore. So we're going to look at our fragment of DNA that contains our insulin gene. And we have to isolate a plasmid from our E. coli bacterial cell. And then we cut our fragment with the same restriction enzymes as we use to cut the plasmid. Then we mix them together. And then you can end up with two options. Sometimes the plasmid will fold back on itself when you add the ligase, and sometimes you will have your inserted gene. Over here, now that we have our recombinant DNA mixed with maybe some plasmids that don't have any DNA, any inserted DNA, then we have to get these plasmids into the bacterial cell. And this is through transformation. And for most bacteria, that just involves changing the temperature. It will make the membrane porous, and it will allow that DNA to go into the cell. Now, sometimes the bacteria are going to have an empty plasmid, and sometimes the bacteria will have a plasmid with our gene. The other thing that can happen is sometimes we can have bacterial cells that didn't take up a plasmid. So we have three scenarios here. And then we can grow our bacteria overnight, but now we need to take advantage of our selectable markers. So we've cut our DNA, we've transformed our bacteria, and we've grown our bacteria overnight. Now we have millions of bacterial cells. So now how do we figure out which ones have our gene of interest? We're going to use two selectable markers. One is the antibiotic resistance gene. So remember, that is only found on the plasmid. So the bacterial population that did not take up a plasmid, they will die on the plate that contains antibiotics. So only bacteria that have a plasmid can survive. Now, after you grow it on the plate, now we have two populations. So now we have some bacteria that have your gene, and some bacteria that the plasmid just reclosed on itself or religated to itself, and it doesn't have the gene. How do we select for those? Now we're going to go back to our LAC-Z selectable marker. So remember our plasmid vector, the multiple cloning site, is within the LAC-Z gene. When there is no gene inserted and interrupting that LAC-Z gene, if you feed the bacteria Xgal, this sort of fake lactose, it will make the colonies turn blue. But what will happen to that gene if we insert, like a transposon, a large sequence of DNA inserting into the gene, you make a non-functional protein. And that means the bacteria can't turn blue. So they will be white. So that is our second selectable marker, and we can end up with a plate of bacteria where we have all kinds of colonies of bacteria. Some of them are blue and some of them are white. So on this plate of antibiotics, let's say we put ampicillin on this plate of agar, which is just a jelly kind of substance that bacteria like to grow on. Any of the bacteria here that are growing have to have a plasmid, because the ampicillin resistance gene is on the plasmid. Any of these organisms that are white will contain the DNA insert. So this is how we can select from these three different possibilities. When we first created our plasmid and then we transform the bacteria, we might have some bacteria that have our gene of interest. We might have some bacteria that have a plasmid, but they don't have our DNA. And then we might have some bacteria that don't take up a plasmid at all. So then we use our selection and our screening methods. If they have a plasmid, they will grow on the antibiotics. And then we look at our white and our blue colonies. Which colonies do we want? We want the white ones. The white ones contain recombinant DNA and disrupt the LAC-Z gene so they cannot turn blue. So now we've cloned a gene. Now we have bacterial colonies that are growing our insulin gene. Now you can make millions and millions of copies. You know, because it has a promoter that is going to be transcribed and translated. And then you can extract all of that insulin protein from the bacterial colonies, and we can use that to treat humans that have diabetes using a human insulin gene. So there are a bunch of different kinds of medications that we can make this way. So here is a chart showing some of the human medications that are made by bacteria.